I have one while you're looking for the next question. I
Okay.
have a question here. It says, when should a team stop focusing on target selection and reevaluate whether the chemical matter itself is the real bottleneck?
That's usually the realm of chemists. I can recall in industry working, you will get your list of hits from a screen, and the first people to see it aren't biologists but chemists, and they'll sit there and say, "Not this one, not that one, not," because they have to be tractable, even though perhaps they're active. Uh, Chemical tractability is very important. So that's usually the first criteria if you have a selection of scaffolds that are active. So you choose that and then see what's active. Obviously, you could order your hits or your compounds of interest in order of potency, selectivity. So you do that as well. What I've been in programs where actually quite early on, you will look at ADME as well. There's no sense in working on a scaffold that's active but has terrible ADME. And you'll probably pick up some catastrophic early safety issues. I recall when we had our first hits for the HIV entry inhibitors, we checked our scaffold on hERG, and it was active on the hERG channel. Well, that's it. That's the kiss of death. No matter how active the compound is, you can't move forward. So the chemists stopped working on activity and built the hERG out of the scaffold first, and then carried on. And so there's a few obvious safety hurdles. You might look at ADME. Potency can be overrated. If you read that there's some tremendous series of articles by Bob Copeland, who stresses kinetics could be more important in vivo than potency. And the general feeling now is potency is overrated. What matters is residence time. So you might also early on look at what scaffolds have a longer residence time on your target. That can also help you zero in on scaffolds of interest.
No, I agree with you. The way I think about it is that we have the ability to run all of these assays. We have the chemists who can look at the compound, and it's a matter of timing and also budget to figure out what's the best order in which these assays need to be run, whether it's a kinetic assay, whether it's on using low expressor versus high expressors, or diversifying the signaling pathway that we're using, or even just at the very beginning include basic ADME in order to sort out the molecules that we don't want to spend time and effort on.
Yeah, and a common phrase that we use that I've seen it elsewhere is fail early, fail fast. What that means is don't waste time on scaffolds that will fail later. You've just wasted effort. Try to pick out those compounds early on and get them out of there so you can concentrate on good ones.
I absolutely agree. And maybe if I can add another layer to that, I think you need to be able to visualize the data. You need to be able to have an infrastructure that allows you to get all of those snippets together. So very quickly you can see side by side, oh, this one doesn't work. Therefore, for XYZ liabilities, therefore we are going to trash it as soon as possible.
That's a good point, because in industry, the industry as I've been in, Early on, you bring in IT, and they construct systems that bring data. When we were at GSK, we had data coming from Britain, USA, Italy, Singapore, and you had to have some place where chemists could see all of that at once. And so, very important to have that. Yeah.
Yeah, and I think it's almost as important as starting to run ADME assays early on with the best hits you have, because having data in different Excel sheets or in different places and not being able, one, as the biologist, to curate them and tell the chemist what they need to be looking at, and then to allowing the chemist and the entire teams to access that data. I think it's that's where a lot of time goes.
Yeah, yeah.